There are estimated to be 5,000 to 6,000 viruses ready to jump from animals to humans,
Nice.
1.5 million unknown viruses out there. The risk of infectious disease is growing as humans increasingly encroach upon the natural environment. These viruses threaten us with the potential of future pandemics, many of which may be more lethal than any we've seen before. Vaccines are our first line of defense against viruses that threaten society. We are currently seeing the severe economic and sociological impacts that a virus can cause in the absence of a viable vaccine. The SARS-CoV-2 virus that is causing the COVID-19 pandemic is devastating on a global level. This is an RNA virus that uses a spike protein on its surface to bind the human ACE2 receptor in order to infect host cells. Moderna, Imperial College in the United Kingdom, and other biotech companies are developing the first mRNA vaccine in response to the COVID-19 pandemic. These vaccines are composed of the mRNA that codes for the SARS-CoV-2 viral spike protein, and once injected into the body, they will be transcribed into immunogenic spike proteins that prime the immune system to attack the virus during future infections. This new type of vaccine has passed initial clinical trials with promising results. Conventional vaccine development takes 10 to 15 years and upwards of $100 million, but mRNA vaccines could be ready in a fraction of the time and cost. However, it's too slow to start development after an infectious disease has already hit. Moreover, as viruses evolve, vaccines can be rendered ineffective, costing lives and resources in the wait for another vaccine. To address this problem, our team created VPRE, the viral predictor for mRNA evolution, an open source software tool that simulates evolution to predict genetic mutations in SARS-CoV-2. VPRE aims to predict the genetic sequences that will likely arise in the future by inferring patterns of evolution from the large dataset of already available viral genomic sequences. These predicted sequences can be directly used to inform mRNA vaccine development by elucidating which regions of the virus are more likely to remain conserved for long periods of time versus regions that are prone to mutation. This would ensure that vaccines being developed have long-lasting efficacy by targeting only viral proteins that are unlikely to change. As our software model is refined and more viral genomes are added to its dataset, it may become accurate enough that its predicted viral sequences can be made directly into mRNA vaccines, giving us a head start in vaccine development before the new viral strains have even emerged. There have been many efforts to model SARS-CoV-2 evolution using traditional bioinformatic approaches, but we chose a novel path of using deep learning combined with time series Gaussian process regression to model and predict how spike proteins are likely to evolve in the coming months. We hope this method is better to capture mutation patterns in their genomic contexts, and also provide insight on how methods like this compare to more traditional approaches. From the GenBank database, we downloaded more than 9,000 SARS-CoV-2 genomic sequences and their associated metadata to serve as our dataset. The first step of our project was to analyze mutations and phylogenetic relationships in our dataset. To look at diversity in the spike protein of these viruses, we studied mutations and their frequencies and constructed a sequence profile, where we determined the probability of each position along the spike protein being substituted by each amino acid. Then we built a phylogenetic tree and grouped the sequences into clades to inform later analyses. Along the bottom of this diagram is our workflow for evolution modeling. VPRE uses a type of neural network called a variational autoencoder, or VAE, to encode viral genome sequences into a handful of numeric variables. The VAE is made up of an encoder and a decoder. The encoder compresses a spike protein sequences into 3 coordinates, where sequences that are similar are encoded into more similar coordinates. The decoder takes 3 coordinates and reconstructs the viral sequence. Because the VAE is a neural network, it needs to be trained. This is a supervised process where each sequence is fed into the encoder, which generates a 3-coordinate representation, which is then passed into a decoder to reconstruct the sequence. Training performance is measured by the difference between the original sequence and the reconstructed one. We realized we did not have enough unique sequences to train the machine learning model properly. To remedy this, we injected spike-like sequences to boost our dataset, based on the sequence profile and results of our diversity analysis. After the VAE was trained, we passed our dataset of 9,000 sequences through the encoder, yielding our whole dataset to 3 coordinates. We then plotted each coordinate against time and modeled the trajectories of these variables using a Gaussian process, or a GP. A Gaussian process is a Bayesian statistical framework for fitting functions to a dataset. In this case, the fitted function simulates the trajectory of SARS-CoV-2 evolution that has happened thus far in the context of the 3 encoded variables. Then the model generates predictions on the 3 coordinates along the evolutionary trajectory. These predictions are passed through the VAE decoder, yielding our predicted viral spike sequences.
We are excited to show some of our early results. From diversity analysis, we tracked down regions on the spike protein that no alteration was seen throughout our data. Right in front of you is a diversity profile of the receptor binding domain. As we can see, we have several regions in green, which means no mutation at those positions is found from our data. Red regions are positions where we see at least one variant. Moving on to the second part. On the upper right corner figure, we're looking at the VAE translated viruses in 3-dimensional space. Each dot is a spike protein. Each axis represents a coordinate translated by our encoder. Colors represent the relative time that a viral protein was detected. From yellow to dark purple, yellow means former and purple means recent in time. Also, we stratified our sequences into training and testing sets based on time, which are represented by dots and triangles respectively. There does not seem to be a clear correlation between the 3 encoded variables from the training and testing sets and progression in time. In Figure 4, we select the viruses from a phylogenetic clade. We then plot each coordinate translated by the VAE against time and use GP to model the trajectory. Green dots represent observed viruses and purple lines are GP regressions. To predict future occurrences as a proof of concept, we sampled 1,000 predictions at 30 days after the last training time point and translated the numerical values back into amino acid sequences. In this figure, 2 most probable predictions are shown in red. Moving down to Figure 5, we showed a fraction of both predictions. It's worth noting that both sequences correspond to an actual spike protein sequence that show up later in time. The only difference between the 2 predictions is one acid, which is printed in bold. Both predictions are associated with a frequency score. As mentioned earlier, we sampled 1,000 predicted values, and since numerical predictions from GP are continuous and protein sequences are discrete, multiple predictions could be mapped back to the same sequence. And that's how we get the frequency score. The top one is predicted to have a higher probability of occurring, and thereafter we compared it to sequences in our testing set, which showed up later in time. If you look at Figure 6, we compare our most probable predictions to sequences in our testing set. As we can see, almost 80% of testing sequences are identical to our predictions. In the future, we would like to further validate our model both in silico and in vitro. As for in silico testing, we would like to model the 3-dimensional structure of the predicted sequence using software like Swiss Model, and use protein docking software like Rosetta Dock to evaluate the predicted sequence binding interaction with the ACE2 receptor. As for in vitro testing, we plan to use a yeast surface display platform and fluorescence-activated cell sorting to quantify the expression of our predicted sequence and their binding affinity relative to the wild-type SARS-CoV-2. Moving forward, we will also need to improve our time series GP model by implementing a multivariate GP regression to capture interactions among the 3 coordinates. In the distant future, we aim to expand the network for other viruses. To end our presentation, we would like to thank all of our advisors who helped us in the development of Viper and supported the growth of our team. We would also like to thank the various and citizens we worked with and talked to in order to improve our project. Lastly, we'd like to thank Dr. Stephen Hallam for overseeing our team and giving us invaluable advice throughout all of our endeavors.
Thank you guys for listening.
Okay. So thank you guys for listening.
All right, so that's certainly a very.
Do you guys have any
Do we
questions?
have any questions?
It's certainly a very timely, project, and thank you guys for doing it computationally, so that we don't have to worry about biosafety and biosecurity concerns. Those two amino acids you were talking about, the two mutations that were of higher frequency, were those located where you would envision the spike binding the ACE2, or was it somewhere else?
It's actually a region that right next to the receptor binding domain, but that region is shown to have the highest mutation rate throughout the data set and also in other, not just within our data set, but also in the global trend, that is the hotspot mutation seen in the spike protein. We looked into that position, so at the original strain occurred in Wuhan, so at that position, it was amino acid D, and then the new strain occurred in Europe back in like April, and then become a dominant one.
Okay. You had talked about when you downloaded all the sequence data, you had mentioned that you also downloaded the metadata, right? In your longer 20-minute video, I think you had mentioned taking into account socioeconomic disparities as far as dispersing the vaccine goes. I'm wondering if you had if you saw any type of correlation between socioeconomic locations versus mutation rate or frequency.
I can maybe speak to that a little bit. I think that was something that we wanted to incorporate into sort of a more robust future model. That we wanted to incorporate sort of a weighting matrix for these sequences based on how frequent they were, so how often they appeared, and eventually give the machine learning algorithm the ability to actually sort of take the sequences individually and give them a relevance in terms of how fast they spread. Because the goal was to track mutations, it was more important about knowing how many people each strain infected, because that way we could, in some way, quantify their capability, let's say, the word is leaving me right now, their viability is the one.
All right, cool. Were there particularly obvious locations that had a lack of data, that had a lack of sequence data?
I don't know that we were able to. Well, we very well could have looked into that, but I don't think we did. At the time, we were mainly focused on the sequences and the changes that we were seeing, because it was all going on over time, and we wanted to make sure that each sequence was unique. We weren't really able to give it that sort of weighting.
But I just want to add, part of our future analysis plans with the phylogenetic tree and the clades is to possibly try to trace which clade is in which geographic region generally, and then try to trace how it spreads that way.
Okay, interesting.
There's a question in the chat. How easy is it to apply Viper model on other emergent viruses? Does it need many adjustments each time you change?
I can speak to that. So I think there were two parts of things that we need to change. So first one is our variational autoencoder. Then they need to recognize other viruses as opposed to SARS-CoV-2. So we need to retrain our variational autoencoder to capture the features from other proteins from other viruses. Then the second part would be for the Gaussian process model. Then the trajectory of different viruses of different viral proteins would be different. And that's why we also need to model that differently, adapting to the data we see from other viruses. but the framework is generally applicable to different viruses.
And it might actually work better for other viruses because COVID-19 or SARS-CoV-2 is a fairly new virus, and there's not, even though there are 9,000 in our data set, there isn't that much data compared to other viruses like the flu or like HIV. So training on other viruses might actually result in a better model.
It's also worth noting that compared to a lot of other viruses, SARS-CoV-2 is quite stable. It doesn't really mutate at the rates that we see in things like influenza. So this type of model, something similar, has been applied to influenza before, and it produced some compelling results.
So with the lack of current sequence data, as far as the lack of number of unique sequences, I guess, how many sequences do you envision you would need to adequately train your model?
So currently in our data set, there are 2,000 sequences that are unique, but we simulated 20,000 spike protein-like sequences to train our model.
Mm-hmm. I can speak to that a little bit. The training of the variational autoencoder, because the way it works is it will essentially streamline itself until it is producing a perfect one-to-one lossless translation of your data set, or as close to it as possible. But the issue with that is then it becomes very bad at describing anything other than exactly what's in your data set. And so there's a little bit of loss in the autoencoder going back and forth. And so to train it properly so that it would pick up the nuances in the sequence, we generated 20,000 sequences based on the sequence profile of our actual data set. And so the divergence in these sequences is fairly low. So if there is an amino acid mutation, we might expect to see it between 10 and 50 times in our 95,000 sequence set. And so because of that rarity, I decided to alter the probability matrix that was generated by our sequence profile and actually change it so that every different amino acid that was represented in our set had an equal chance of appearing in our training set. And what that did is it increased the resolution of the autoencoder so that it was able to pick up the nuance changes in our actual data set. And so if you did something similar for other viruses, you could expect that it would train the model similarly to what we achieved. The real thing that we're waiting for, and the reason that it worked better with influenza, is just the magnitude of the mutations is so much greater. There's so many more changes happening year to year and plus we don't really have the year-to-year changes for this. So it's it is more active in mutation, and it's also that we just don't have enough data. And to speak to your question as to how much we would need, that's just a really difficult question to answer because of the fact that it doesn't mutate as frequently as our experience with previous viruses.
So being a predictive model, do you have an idea of what kind of time frame we're talking about? How far out can we predict? Is that a valid question?
Well, we are obviously still verifying our predictions. And so I don't want to say we can for sure predict anything with a lot of reliability. But right now our model is set to predict about 30 days out at last time. Amelia, is that right?
Yes, that's correct. So as a proof of concept, we just sample the 30 days after the last time point in our training set.
Yeah, I think.
Yeah.
Sorry, you go.
Oh, go on,
in order to answer that question, we would
No.
have to go through some rigorous backtesting. So we would make predictions, and then we would have to backtest and see if our predictions were actually accurate before we could know for certain that we're predicting anything with accuracy. I think there's a question in the chat as well.
So the question says, do you think that if the mutations and potential evolutions you found occur in the common COVID strain, that this would reduce the effectiveness of a vaccine developed before those mutations occurred?
It depends where the vaccines are actually targeting. So if the protein in which, if the protein that the vaccines are targeting for immunogenic purposes happens to mutate, and the antibodies we produced for that vaccine can't cross-recognize to the mutated protein, then that would cause a problem. There might be less effectiveness to the vaccine, but it really depends where the mutation is and what the vaccine is targeting.
Yeah. Also, generally speaking, we did find that the spike proteins were very highly conserved. And so just generally speaking, it's likely that any vaccine would be, like I have been saying, it does mutate a lot less frequently than influenza. So as we're seeing sort of seasonal influenza vaccines, I think we would expect it to be even less frequent with SARS-CoV-2. But I am definitely not an epidemiologist or a virologist, so I can't speak to any of that with any certainty.
Interesting. There have been a couple papers saying that the SARS-CoV-2 virus has mutated, and that's changed which strain is dominant in the general population. So that might be an interesting thing to look into if you're into that.
Yeah, and also to add to that is a lot of the vaccine companies are targeting the spike proteins for vaccine development.
So I have kind of a related question. And the question is, like, assuming your model had, let's say, just for the sake of the conversation, say you had like perfect predictive power and you could predict what the trajectory of the evolution is going to be. Like, where do you see such a model kind of fitting into like a drug development pipeline or something else to combat the COVID-19 virus? Like, where do you see this model having utility?
Well, I can perhaps speak to that a little bit. I mean, it is feasible to have predicted proteins synthesized. These proteins aren't particularly long, 12,073 amino acids long, and so I'm personally unaware of the folding effects that are necessary for these proteins, but to have the viral RNA modified in such a way that they were represented on the protein surface isn't unthinkable. And so our thought was that it could be generated in a lab and tested with the vaccine to ensure that efficacy was conserved.
I think there are generally...
And also...
Oh, sorry. Go ahead.
Okay, go ahead. Okay, so I think lots of the vaccine company is answering one question: will our vaccine work, and how long our vaccine will be effective? So a model like ours would be able to help the vaccine company to estimate whether this virus would mutate such that the vaccine they generate will no longer be effective. Then they need to develop newer vaccines to make sure that people will get a higher quality vaccine at a time in the future.
Yeah, just to summarize, so I think there are two routes there. One is to predict what mutations will happen so that we don't target the regions that the mutations will happen in. And the other thing is that if our model is confident enough, we can actually, well, theoretically, we might actually be able to take those spike proteins and then translate them directly into mRNA vaccines, because it's just the genomic sequence and then straight to mRNA. And mRNA vaccines have gained a lot of ground in the past couple of years. So that was something we were thinking about that might be possible in the future.
Thanks for your answer.
So do you have enough? Sorry, go ahead. Oh no. Do you have enough? Do you have enough data to run through Viper to be able to benchmark that accuracy against other predictive algorithms out there?
Are you referring to predictive algorithms regarding SARS-CoV, or are you predict other algorithms for other viruses?
Let's talk about COVID-2. Like, I think as far as your HP is concerned, I think you interviewed at least 3 people. I think one of them was a professor working on COVID-2 evolution, right? So, did your results look anything similar to his, or how did that comparison look?
I think.
Ariel, can you speak?
Yeah, that's I think
Sarah?
you're mentioning
Yeah,
Dr. Stranadka that we talked to at UBC. So her team is working on something similar. But the last time we spoke to them, they were still at the wet lab stages. So they were kind of modeling in vitro spike proteins and then gathering, what do you call it? Like interaction data between the in the folding physics of those proteins. So they weren't quite at the predictive stage yet, and we weren't able to compare. We were just asking them for guidance and information. Yeah, so we don't have that benchmarking data for our software yet.
Yeah.
And also the true hallmark, or like the true test, is going to be validation of sequences that haven't been seen before that have been generated in our algorithm. The comparison of predictive power can only really be validated against the accuracy of the predictions.
Sure, true.
I have a human practices related question. So just taking a look at your webpage there, just with all the cool stuff that you guys have been up to with your project, outside of the lab, everything from the children's book to the online curriculum series, like as an educator in the public, it makes me happy to see these initiatives rolling along with what you're doing in the lab. I was just kind of curious about how this impacted your work going forward, because reflexivity is a really huge thing with education, especially when you're engaging with the public. So I'm just curious, from talking with your stakeholders, from seeing the reactions to your work, how has this informed your future practices on this side of things?
Yeah, I think the approach we took with practices this year was kind of targeting different age groups. So you can see we're targeting very young children with the storybook. With COVID, there's this increased or heightened awareness of, hygienes and hand washing. So that's something that we wanted to, bring about as a moral of the story. We also, do interviews, and we talk with ages through our written video series, where we kind of just discuss synthetic biology in general with people of different ages. And it's always nice to kind of hear their reflections and to see what they think about synthetic biology. We also did an infographic on COVID-19. There was different sections in it, but one of the sections was, debunking the myths of COVID-19 and things like that. So I think overall, we're just trying to increase communication about this field of science. And we also interviewed university students about what they think about synthetic biology and their knowledge around it, software and things like that. And we realize sometimes there's misconceptions and things like that. So overall, our initiatives through human practices was kind of to address those misconceptions and just kind of increase knowledge around this field.
Awesome, thank you.
Mm-hmm.
Sorry, my Wi-Fi cut out there for a moment. But to add on to that, I think something we've come to appreciate a lot doing this project is open science. And because we were able to get these giant data sets basically for free, like open access on the internet, we would really like to contribute back to that. So this is why our software is open source, which is why we're making such an effort to put all of our methods out there, and then also to develop kind of symbio curriculums so that we can give it back to the future population of university students and people who want to learn about these kind of things.
Just to add on, because this year we didn't do a wet lab project and we're doing like a bioinformatics workshop, a bioinformatics project, we actually created a workshop on bioinformatics just to, improve understanding around this topic. It's very beginner oriented, but I think it really helps, people to, get started and learning about bioinformatics. So within our workshop, we had a segment just to help people learn about VPRE and just kind of the concept behind it, the predictive aspect of VPRE. We had like an analogy activity to help people kind of conceptualize and understand what VPRE is trying to do with the predictions. So I think this is a really valuable initiative that we've been pushing with human practices.
Beautiful, thank you.
Yeah, so maybe tell us a little bit more about your collaboration opportunities, if you would.
Ariel, do you want to speak to that?
Collaboration with other iGEM teams or just?
Within your own team, with other teams.
Yeah, I think I could speak to this with regards to integrated human practices. We're always trying to, get to interview people to really help develop our project. So we have spoken to, for example, I think you asked earlier a question about, developing countries and things like that. So we did speak to an economic professor in Peru about, how is the vaccine program there, and how easy is it to disseminate mRNA vaccines there. And we learned that over there, medical sciences facilities is not as well evolved and well funded as in North America. So it is definitely a challenge over there for opportunities in science and research. But we also learned that there is actually a solid vaccine program in Peru for pregnant women and for children, but less so for adults and seniors. But I think that the thing that we got from it was that, there is already a framework in place, and that, we're not starting from zero. If we were to disseminate, an mRNA vaccine or something like that down the road, we're able to build off of something. So it's really valuable to learn from, a developing country point of view, and just to really consider not only, the North American population, but to consider some developing countries or marginalized communities. Yeah.
Sure.
There's also an opportunity. So we did reach out to Eric Ma, who was the one I spoke to you about the influenza, the application of this sort of technology to influenza that we had talked about before. And we reached out to him to see if he had any experience or knew anything about taking a more multivariate, interdependent approach to this model. He said, okay, I'll get back to you, give me a few days. And he went away and came back and said, no one is doing this. This is the edge of human knowledge. And we were like, okay, so working with somebody who is developing that sort of technology, who is really interested in that, would be something that we were interested in. We looked around for it, and it isn't out there right now. So, or at least if it is, it's a well-concealed secret. But Eric was able to give us that, at least, that it's that we're not fools for not being able to figure it out. It literally doesn't exist, which was comforting to all.
Nice. So how about other teams? Have other teams helped you out with any of this, or have you guys been able to help other teams with their projects as well?
I think back in July we attended. Back in July we attended one of the conference held by Calgary. It's called iGEM. So we presented our preliminary project idea there and received some feedbacks from the team and also the professors from the university. That sort of guide us how we present our project, how we communicate with others.
Yeah,
kind of
and also just to add on, we also spoke very early on in the summer with some grad students that were overseeing the iGEM team at U of T. Because at that time we were piloting our synthetic biology curriculum, and we weren't too sure how to approach it because it's such a big scope, right, that we're trying to tackle. So we spoke with them, and they gave us the idea of interviewing local UBC university students in science, and just kind of hear what they think and what they know about synthetic biology and what they may be understanding incorrectly and things like that. So I think that was a really nice meeting that we had with them, just to get our ideas started. And we did indeed implement some of their suggestions, and we interviewed like 15 university students from UBC that are in first or second year science to hear about their knowledge about synthetic biology and so on. And I think that really kickstarted our synthetic biology curriculum, and we really based our curriculum off of their feedback and things that they wanted to learn.
Okay, I see. Nice. So, Austin, if you have anything else.
Yeah, just an amazing project, guys. Really, really good job. Really.
Nothing for me. I don't know if any of the judges have any questions left.
I think I'm good as well. What year are you guys?
A bit of a mix. I'm in my 4th year. I'm graduating in April.
Four.
Fourth,
But
yes.
on the team, we have various different year levels. We have students from 1st year. We also have students like going into their 5th year, are ready to graduate next year. And we also have a very diverse demographic on the team. We have engineers, we have computer scientists, as well as microbiologists, biologists, virology students. So it's a very, very interdisciplinary team.
Okay, good to know.
Yeah, I'm actually a chemical and biological engineering student. So when I said I wasn't a virologist, I really meant it.
All right, well, good to know that you guys have an interdisciplinary team there.
Are there any more questions?
I think I'm good.
And there's, oh, there are 2 minutes left.
Sorry, Austin, you were speaking before. I believe you got cut
off.
Oh
yeah, no, I just wanted to continue showering you guys with praise. I just, you know, it was a tough year for everybody, but, you know, you guys did amazing with, you know, making the best of it, you know, doing something online, research exclusively, totally ambitious, but you guys rocked it. So great job, truly.
Thank you so much.
Thank you.
Fatima says she likes the SciComm level on the website. And thank you so much for that.
Sorry if I cut Austin off earlier. I'm realizing I can only hear like half of the people in here. I just noticed Austin's mic was green, but I couldn't hear a word he was saying. So apologies if I've been like cutting somebody off this whole time.
No, it's okay. It's happened. A few people have done it a few times. I think it's just a communication thing. None of us blame you. It's all good. Okay.
Well,
Okay,
yeah,
that's probably time, but thank you so much for listening, everyone. And for all the feedback as well.
Thank you so much for your feedbacks and all the questions. And hope you guys enjoyed our presentation.
Yeah.
All right, well, thank you guys as well. Yeah.
Thank you.
Bye.
All right, then.
Bye-bye.
Okay, bye.
Bye.